So I was going to read the introduction to our speaker today, and my boss told me that I could not read the introduction. So I'm going to introduce her as my friend, which we are, and it's a pleasure to have Katalin Szaszi working with us here at St. Mike's Hospital. She is an associate professor in the Department of Surgery. She is a scientist here at Kidney Research Center. She has her own lab, so for the high school students that in the future will want to do research, it's a great opportunity to talk to Dr. Szaszi and get the stage set up for future endeavors. Her main area of research is on inflammatory response, basically looking at inflammatory response to kidney injuries. So when we have a patient who sustains trauma, for example, a guy gets shot here at Eaton Center, and there's a major injury to a major vessel, so the patient bleeds a lot. What our body does is to shut down all the vessels so that the patient, number one, does not lose any more blood, correct? You get vasoconstriction. And number two, the remaining blood that's in our system is diverted to noble organs, we say, the most important organs, which are the brain, the heart, and the lungs. So we forget about the kidneys. We forget about the gut. And the time that these organs, the kidneys and the gut mainly, go under ischemia or under low perfusion, results in a major inflammatory response. 80% of the time after the surgeon operates on the patient, stops the bleeding, the patient is taken back to the ICU. Usually they spend a few weeks in the ICU. 80% of the time, those organs, particularly the kidney, regains its function again. But in some patients, that doesn't happen. The main cause of all this is a very intense inflammatory response, which destroys the cells of the kidney, and to a point where it's not recoverable anymore. So this is what's the main topic of this talk today with Dr. Szaszi, will be to how can we probably modify this in the future. So she studies the inflammatory response and how can we either stop it or make sure that the organs recuperate their function after stress like this one. Okay, so I'm happy to introduce Katalin. Thank you very much.
Thank you very much.
And we'll be honored to have to hear her talk.
Thank you very much for this wonderful introduction, and thank you very much for coming, and really appreciate that you're here. Okay, I see that I need to talk more to I have to solve the problem of being visible behind this stage and to still be able to talk into the microphone. Is that better? Okay. So basically, thank you very much for coming. It's snowy, it's cold, and you're still here, so I really appreciate that. So what I'm doing in this research institute, I'm trying to figure out how cells work. I'm a cell biologist. And you know that cells are basically the building blocks of all tissues and all organs. So you can't really understand how an organ works unless you understand how a cell works. And I think that cells are really interesting and really wonderful, and I will talk to you about some of the cell biology of the kidney, because the kidney is my main organ of interest. So my presentation is going to be divided into 4 sections. First, I will give you a little refresher about the kidney. I will tell you how the kidneys work, what are the most important things that we should remember about the kidney function. Then I will talk to you about how kidney function can go wrong, and I will introduce to you to some kidney diseases. And then I will get to the cells of my interest, which are the tubular cells. And finally, I will give you an explanation to my title. What are gates and what are fences? Okay? So as you all know, we have two kidneys, and ideally they look beautifully like this. They are shiny and they are a very symmetrical organ, and they are absolutely vital for the normal functions of the body. Why is that? That is because the kidneys basically operate like a little water cleaning organ. They eliminate excess water from the body, and they make sure that we only have the products that we need, and we get rid of the waste products that our body produces. So you may remember this from your biology studies. So the main unit of kidney function is called the nephron. And the nephron really is composed of a beautiful organ at the beginning, which is called the glomerulus, which I will talk to you about in a second, and a tubing system. Really, that is the plumbing system of the functional unit of the kidney. We have approximately 1 million nephrons in our kidneys to start with, but as we grow older, all of us are going to be losing some of this. Hopefully not too many. And if we lose too many, then the kidney will not be able to work properly, and that's when we will end up with kidney diseases. So let me talk to you a little bit about the glomerulus. So basically how a nephron starts is with a glomerulus, and this is a tuft of capillary cells, capillaries. So the blood vessel comes into this little organ, and it divides into many, many capillaries, and these basically make up a large surface where the filtration process can take place. So what is the filtration process? When blood enters, it is filtered through the walls of this capillary, and a lot of water, a lot of ions, but not large proteins are going to be getting into this filtrate. Now, let me show you, because this is really worth looking at, how this little apparatus looks like. So this is a scanning electron microscopic image of the glomeruli. This preparation was specifically prepared in order to show us the blood vessels. So you can beautifully see that the glomeruli are really composed of a tuft of blood vessels. And this image on the other side shows you once again a scanning electron microscopic picture, but now of the full glomerulus. And within these section there is a blood vessel, but the blood vessel is covered by these wonderful processes which come from a cell that's also within the capillary. So basically this structure is made up in such a way that the capillary wall is covered by these processes, and you can see that there is a little space between these processes. That's when the filtrate can get out from the capillaries. You can appreciate that this is a really interesting and fine structure, and because it's such a fine structure, you can also appreciate that a lot of things can injure this structure. And once this beautiful architect of the glomerulus is injured, that's when disease processes start. And I will get back to that in a second. So once the filtrate is generated, it gets into this tube system. And it's moving all the way through this really large and really convoluted tube system. And what happens in this tube system is that things are taken out from the filtrate and things are added into it. A lot of water is actually taken out from the filtrate. So you generate about 180 liters of filtrate every day, but you end up urinating only about 1.5 liters of urine. So you can appreciate that really a large amount of water has to be taken back. But there's also a lot of ions that needs to be added or taken out. So the final adjustment of the composition happens in these tubular structures. And in the end, what we generate as the final urine is going to be much less in volume and is going to be adjusted according to the body's need. So we're getting rid of whatever we don't need, and we are making sure that we're not losing things that we need, such as sugar, such as proteins. Those are things that we should not be losing in the urine. Okay, so what happens when things are going wrong? And there was one example in the introduction that is a very dramatic example when there's a trauma and the kidneys simply don't have enough blood flow to them. That can destroy the kidneys because they need a lot of blood flow in order to function. But very common, and unfortunately way too common, is a problem associated with diabetes or high blood pressure. These are the two most common diseases that affect the kidney. And you know that diabetes especially is basically an epidemic. There's really way too many people having diabetes, and unfortunately the kidneys are going to be affected by diabetes. Now, the good news is that this is happening slowly. The bad news is that this is a process which we don't know how to stop or revert. So really, research is needed for us to understand much better what's going on. Now, this little scheme was, they call it the diabetic ribbon, was put together in order to help people remember and understand and look up what the symptoms are of diabetes. So it's called the diabetic ribbon. You can see that it lists the symptoms. And on this side, you can actually see that one of the earliest symptoms of diabetes is going to the loo all the time, especially at night. So this right away implies that there is something wrong with the kidneys in diabetes. And indeed, what happens in diabetes is that the body is unable to handle sugar. Now, glucose, a type of sugar, is supposed to be highly regulated in the blood. And in the case of diabetes, it's not regulated anymore, so the person has high levels of glucose, and this gets into the filtrate. So now you end up with a lot of glucose, and glucose within the filtrate is going to disturb the fine transport processes of the tubules. So most importantly, what happens is that glucose doesn't allow the tubules to absorb, to take back water. So you end up with a much larger amount of urine, and that has a lot of consequences for your well-being. On the other hand, albumin, a protein which is present in the blood, is not supposed to show up in the urine ever. Unfortunately, diabetes destroys the nice filtrating function of the glomerulus, and because the filtrating function of the glomerulus is faulty, now you end up with albumin within the filtrate. So what happens? Why is this a problem? It's a problem because, again, it disturbs the transport functions of the tubules. So the tubules are now not able to adjust the composition of the filtrate. But even worse than that, the presence of glucose and albumin actually affects the cells which are lining the tubules. And eventually, the presence of glucose and albumin will destroy the cells in the tubules. So now you end up with a dysfunctional nephron, and eventually you lose your nephrons. And as I said, this is a progressive disease. We really can't stop and can't turn it back. And what you end up with is, instead of a healthy, nice kidney, you've got a diseased kidney which is much smaller. You can appreciate that it's not as nice and not as shiny. And the reason for this is that the healthy tissue, the healthy cells are now taken over by scar tissue, and the whole kidney is now showing scar areas. And instead of having healthy nephron, you're having non-functional scar tissue. And in the end, you end up with a so-called end-stage kidney disease, which is not curable. We can treat this disease because dialysis is a good treatment for kidney disease, but the problem is twofold. First of all, people with kidney disease are really not healthy. Even if they are having dialysis, they are really not feeling well. They are very sick. And the second problem is that this is a very expensive treatment way. So the best would be, obviously, is to prevent or find a cure for this disease. And that is why we need to understand exactly what is going on. So this brings me to talking to you about tubular cells. And tubular cells are the cells which line the tubules, and this is a kind of epithelial cell. And epithelial cells, this name is applied to cells which usually line organs, And these cells are sitting closest to the inside of a lumen, or basically make an interface between the external environment and the internal environment. So think of your guts, for example. The guts are lined by epithelial cells. Similarly, the kidney tubules are lined by epithelial cells. And these cells are indeed making a nice tubular structure. And if you look at it with a scanning electron microscope, then you can actually see that indeed there is a tubular substructure. There are individual cells sitting in this tube. But interestingly, they have these fine hairline processes on top of them. This is another representation, a cross-section of the wall. And again, you see these hairline fine processes. These are called microvilli. And the reason why the cells have these is that it makes their surface large. It gives a lot of area through which transport can occur. This is very typical of cells that perform a lot of transport processes. Okay, so what are these cells doing? So the tubular cells, according to their functions, are really built for transport. So what they need to do is to take stuff from the lumen and transport it toward the kidney tissue. And in the tissue, those things are going to go back into the blood. So eventually, whatever is going to be transported for the lumen is going to go back to the blood. And what are those things that are being transported? Well, the tubular cells, at least in some portions of the tubules, can transport glucose. So good news, we can recover some of the glucose that is being lost, because we always lose some glucose. But in case of diabetes, we can't recover all of it. There's a lot of ions that are being transported from one side to the other to adjust the composition. But what's really spectacular is that these cells are not symmetrical. They have different composition on the side facing the lumen and on the side facing the kidney tissue. And why is that? This is because that's basically the basis of a transport process from one side to the other. The process is done by so-called transport proteins. So proteins, for example, that can grab sodium ion and a glucose ion and transport it into the cell. And then on the other side, there's another transporter that can grab glucose and transport it to the blood. But if you had the same transport proteins on two sides, then the transport would be going back and forth. So that's not good. So we need different transporters on one side, and again different transporters on the other side. And this is exactly what happens in the case of epithelial cells. They are non-symmetrical. This is called polarization. So they are polarized. They have a different one composition on one side and a different composition of other side. So this function is this is absolutely important for their functions. Sometimes polarization can go wrong, and there's a very, very interesting inherited disease which is called polycystic kidney disease. You may have heard about it. This is actually not such a rare disease. 1 in every 1,000 babies are thought to be born with this disease. And in fact, this is a collection of diseases, so there are different causes. But what happens is the same in each of these diseases. This is a problem with orientation of the tubular cells. The cells are completely disoriented. In normal situation, they face the lumen with one side, and they face the kidney tissue with the other side, and they have a long process called cilium, which tells them which is one side and which is the other side. Now, genetic problems can actually make this cilium not work properly, and if that happens, the cells are disoriented, and they start facing in each direction. The transport is not happening correctly, and there is fluid that builds up in the tubules, and also the structure of the tubules is completely disrupted. So what happens is that the tubules end up having all kind of cysts, so basically extra little portions that keep enlarging, and the patient will end up with a cystic kidney, which is non-functional. And This picture, it's not beautiful. It shows you that indeed the shiny, beautiful surface of the kidneys is completely disrupted, and there are now a lot of cysts. Okay, so this brings me to the thought of gates and fences. Why can these cells maintain their polarized structure? The thing that maintains their polarized structure is a junctional area, a contact between the two cells, and this is called the tight junction. And the reason why it can maintain the polarization is that it sits at a given area in the cells. I will show you in a second where exactly. It connects the two cells, but it also seals down the two areas of the cell, the one side and the other side. So it basically acts as a fence. It doesn't allow the proteins from this side to be mixed with the other side. But this structure does another very interesting thing. It doesn't allow unrestricted fluid or ion or substance flow between the cells. Imagine if we had a big space between the cells where everything could go through, then we built up a transport process, but we would destroy the transport process because anything could just go between the cells. So we do need to seal that space off. And that's what this structure called tight junctions are doing. So basically what they are gates because they won't let anything unrestricted through. They do let something through, so that is something that I will show you in a second. And they are fences because they don't let the two sides of the cells to mix. And if one looks at epithelial cells, tubular epithelial cells, now these are cross sections of the cells. And let me show you on this picture. This is where the two cells meet. And this is an area where you can see that the two membranes, so these dark lines are the membranes, they are getting close together. And the two cells are really aligning together in a tight structure. So these are the tight junctions. Okay, so tubular cells can be cultured in the lab. And this picture is from cells that have been cultured in the lab. This is actually done by a student in my colleague's, Dr. Kapush's lab. It was done at the U of T scanning electron microscope facility. And you can see that these cells indeed beautifully have this microvillar structure. They are not standing as in many of the pictures because of the fixation process, but indeed their surface is full of these beautiful structures. Moreover, they have this cilium structure that I mentioned to you earlier. This is the structure that tells them how to orient. Okay, so tight junctions really do seal off. And this is a wonderful experiment that shows how the tight junctions are sealing one side from the other. So in this experiment, a tracer molecule was given to one side, and then it was followed using an electron microscope. This tracer molecule is dark. It shows up as dark. But you can see that it doesn't get between the cells. Now if you give it from the other side, from this side, then you can fill up the space up until a point. You can't go with this molecule any further. It is beautifully sealed off. So true, tight junctions do seal off. Now, how can we in the lab measure the transport function? Because the tight junctions do seal off a lot of things, but they do let through some molecules. So you need to use a tracer molecule, which is fluorescently labeled, and add to the cells on one side. If the molecule is small enough, then it will show up on the other side. And you can measure this. So for example, these cells were treated to increase their permeability. You can actually see that indeed there's some molecules measured on the other side. However, if you give large molecules, then you cannot measure, then you cannot detect them. They are not going through these cells. So indeed, tight junctions do let through small molecules, but not large molecules. Through gates, which are acting like a selective gate, some molecules can go, others cannot. So another way for us to culture the cells and to show that they are indeed forming a very tight seal, is to put them into a matrix where they are growing in three-dimensional structures, and they end up making a three-dimensional cyst. So remember this picture that I showed you before? Basically, these cells are now reproducing what happens in the tubes. And the green labels their plasma membrane, their edges. And you can see that indeed, this structure looks very much like this one. This is how the cells were growing. Now we know that within the cells, within this structure, there's no cells, there's a cyst, it's empty. Because if you label the nuclei of the cells, they are surrounding this cyst, but there's nothing inside. Now, if you make this seal, the tight junction seal, more leaky by removing calcium, because that's something that makes the tight junctions very leaky, then you can start showing that things which are added from outside, and the red dye that I show here was added from the outside, starts to show up in the middle. So this red dye is not showing up unless you disrupt the junctions. So we can really show that the cells are forming a seal and that that seal can be manipulated. Okay, so what are these junctions made up? And this was really a mystery for a long, long time. People started looking at these structures in the 50s. The first electron microscope pictures were done, and they started seeing these wonderful structures, these tight opposing membranes, but they didn't know what they were. And the only thing that was clear from the pictures was that the membranes are getting close only at given areas. And these were called kissing points because they didn't know what they were, but they wanted to give them a name. So these kissing points turned out to contain proteins. And nowadays, we know that there's a whole bunch of proteins, over 100 proteins that reside in the plasma membrane. But really, the first one that was found is called occludin. This was the first, and probably it's the most famous tight junction protein to date. So it's a protein which sits in the membrane, has some portion on one side and some portions in the other side. Now, how can one imagine? Actually, let me show you the other proteins. So now we know that there are a whole collection of proteins sitting in these structures. So how can one imagine the way these are sitting in the membrane? So this depicts one side of one cell, and this one depicts the other cell. So the proteins are sitting in the membrane, and parts of these proteins are facing each other. And where they are facing each other, they are actually connecting, making a connection with each other. And this is where the seal can happen. And then they have portions which are connecting to the inside of the cell, which turned out to be almost as exciting and almost as interesting as the portions which are facing each other. Okay, so how can we visualize, how can we show these cells? So for that, we use a technique called immunofluorescence. And that takes advantage of an antibody. And an antibody is a protein produced by the body which is able to recognize specifically a protein. So we use an antibody which is then visualized by a fluorescent second antibody. And then one can look at in the microscope and see what the result of that staining is. And if you do that with tubular cells, for example, to visualize occludin, then you got these beautiful cobblestone-like structures. So now you're looking at the cells from the top. It's a cross-section, and this is one cell, and this is the edge of the cell. This is the second cell, and this is the edge of the cell. So indeed, these structures show up only on the periphery. Okay, so now the second most famous molecule residing in the tight junction, which was discovered much later, is the one that is actually doing the sealing and doing the permeability function. And these proteins are called claudins. And for the sake of time, I'm not going to go into any of the details, but what these claudins are doing, they can form channels through which things can go through, and these channels are highly specific. So there is a high specificity we're letting through only certain ions, only certain substances, but not the others. So if I get back to my original question and my original problem, what happens during a disease? So one can assume that when the tubular cells are having functional issues, then the junctions will also change. And that is exactly the case, which I'm showing you here. If we stain one of the clouding proteins, and again you see these beautiful cobblestone-like structures, and you see the nuclei of the cells, then we treat with an agent that is known to be highly present in various diseases. It's called TNF-alpha. And you can appreciate that short-term treatment or a long-term treatment will really change how these structures look like. So these are the kind of experiments that one can do to find out what exactly happens and what exactly is going to be the cause of these changes, and how are we going to interfere with these changes, because eventually what we want to do is interfere with these changes. Okay, and then finally, just to show you, and this is my final slide really, just to show you that these cells are not just boringly sitting there and transporting molecules, they can actually move. And this, what you see here, is a layer of tubular cells which were cultured in the lab. And we made a wound right in the middle of it just by scraping away some of the cells. And we wanted to see what happens. How do these cells react? How do they repair themselves? And look at what happens. They start moving. The movie is looped, so you're going to see it again and again and again. And the cells are moving a little bit out of focus. But what you can really appreciate is that they are having contacts towards the back area. They lost their contact toward the front, and that's a stimulus for them to move into the front, but they never lose contact with their bodies back towards the tissue. They always keep contact, and they're basically dragging forward the whole sheet of epithelial cells. And let me just show you this again, if it moves. Fortunately, it moves a little bit because there's something happened with the transfer, but this is a slower movie, and you can see that this front is basically moving until eventually it will reach the other side and the layer has healed itself. Now the very interesting thing is that this process also depends a lot on the junctions between the cells. So this is a very exciting new area, and we're just starting to look into this, and it's probably going to be the basis of understanding how cells and layers repair themselves. And finally, of course, the experiments that I have been showing to you were done by people in my lab, so I would like to thank them for that. And no science can be done without money, so we've been receiving funding from some of these agencies. Thank you very much for listening to me, and I'm happy to answer your questions.
Does anyone have any questions?
The tight junction itself, is it one protein that traverses both membranes, or is it a protein-protein interaction?
So it's a protein, it's a collection of proteins. Oh, sorry, I have to come back here. It's a collection of proteins. So there's a lot of membrane proteins, different types, but the membrane proteins are actually going through the membrane and reaching the other proteins from the other cell. So imagine one cell, and let me show this to you, it's easier to show. So basically, this is the membrane, and this protein has towards one side, to the outside of the cell, some processes, and the other protein will come in with the similar processes, and they make contact. But each junction contains several kinds of these membrane proteins, and I didn't have time to talk about it. On the inside of the cell, there's a collection of cells which are not membrane proteins, collection of proteins which are not membrane proteins, but are binding to this protein. So really, in the end, it's a large collection of proteins. But only some of them will be doing this, meeting the other side.
So I wonder what's making the seal. Are there actual cysteine bridges between them?
So that's an excellent question. Yes, there are some electrostatic interactions between the two proteins, but nobody really knows what makes the seal. So that is a question that we still don't understand. But we know that it depends on these proteins sitting and meeting each other, because if you make changes in the areas which are meeting each other, then the seal is gone. But it's even more interesting than that, because they're not only making seals, some of them make seals, but some of them make channels through which ions can go through. So it's at the same time sealing and making permeability. And it depends on these proteins, but we really don't understand all the details of this. Yes?
Yeah, I was just wondering, you were talking about TNF alpha and how it causes them to break their junctions and move. So now, like, TNF alpha is like from the innate system, it's like a cytokine, right? So now, are there diseases where it's like an autoimmune disease where the body is fighting against the kidney, and then that's what's causing the problem by breaking the tight junctions?
So yeah, thanks for that question. I really didn't have time to talk about too much about TNF alpha. It's a very exciting and interesting cytokine because, as you said, it is a cytokine that has immune functions, but it is a molecule that is released during inflammation. And inflammation really is a response that's given to any kind of injury. So in the end, it is a molecule that is present at any kind of injury. And it was shown to be very important in chronic kidney disease. Actually, diabetic kidney disease depends a lot on the presence of TNF alpha. And we don't really know what exactly is going on with the junctions, but surely they are disrupted because we know that in other organs, TNF alpha disrupts the junctions.
When you show that picture of the both edges coming together after you scraped it between them, so that was a cell culture media, correct? So if we translate that into a diabetic kidney, so you're having injury, why doesn't the injured kidney in a diabetic patient just do the same thing, that both edges come together? Is it because you have cells that will prevent them from reaching both sides?
So that's an excellent question. So the thing is that actually there is, nobody really has been able to look at this process in an intact organ because it's not trivial how to look at it.
Yeah.
But this process is definitely going on because there is some healing going on. And in fact, the problem is that healing and migration of the cells is itself will destroy the intact function of the tubules. So if it's going on for too long, then that is the process through which the scar tissue is generated. And so if you have a little bit too much of the good thing going, yes, the organ tries to repair itself. Probably the tubules are trying to repair themselves. But in the process, the tubular cells are modified in such a way that in the end, if the process is just overwhelming the tubules, they will not come back to be intact tubules anymore. So this process probably, in the end, if there's injury is too big, is contributing to destroying the organ.
Very interesting.
Yes, please.
So, say if a cell lost contact with its neighbor, so would occluding activate downstream signaling somehow to modulate acting dynamics?
So that's an excellent question, and yes, that is exactly what's the most exciting thing about the junctions, is that if they lose the contact with the other cell, then there's a lot of signaling going on, a lot of signals initiated, and one of the things is actually active, yes, that is being regulated. So just the fact that the cells are losing contact with each other, so the proteins like occludin cannot bind to the occludin from the other cell, initiates a process which tries to repair, restore the integrity of the tissue. And that is why probably the repair itself and the migration itself is junction dependent, but we don't know all the details of this. This is a very exciting and a very hot area. Yeah. Yes?
Yeah, there's also familial diseases associated with the kidney disease, right? So I was just wondering if, like, I know cadherins and nectin, they're the ones that attract each other and then signals cascade through the cell. I was wondering if it's like genetics and like that's causing like there to be some problem with the cadherin to the nectin that's causing other types of kidney.
That's another excellent question. So I didn't really have time to talk about those junctions. Those are not part of the tight junctions, but those are part of the so-called adherens junctions. So there are more than one kind of junctions. My interest is in the tight junctions, but the adherens junctions, I have to admit, is almost as exciting as the tight junctions. Catecherins are the molecules, the most important molecules sitting in the in the adherens junctions. And I don't know of any genetic disease affecting the catarins. I'm sure there are some, but what I know is that, for example, when a cell is transformed to become malignant, the caterin levels are changing. So the cells may lose its caterins, and that is part of the transformation process. So it's a very, very important and a very, very interesting question. What happens to caterins during disease states? And cancer formation is really one of the most interesting process where caterins probably have a role. What exactly, we don't know, but when caterins are intact and when the cells are sitting there with their caterins forming junctions, then the cells become quiescent, which means that they don't migrate, they don't proliferate. So that's a very important signal. By the way, tight junctions can do the same. They can stop the cells from migrating and proliferating. So when the junctions are intact, cells are quiescent. So E-caterin levels changes are probably very important for transformation.
More questions? Okay, so I'd like to thank Dr. Szaszi
Thank
for
you very
this
much.
brilliant lecture.